Okay, well, good morning, everyone. Good morning. I'm Marie Diener West, and on behalf of the Johns Hopkins Center for Clinical Trials, I'd like to welcome you to the first seminar of the academic year. And it's my pleasure for this seminar to be able to introduce our speaker, who's Dr. Simon Day. So Dr. Day has spent more than 25 years working in the field of clinical trials, mostly in the pharmaceutical industry, but also including 5 years at the UK and European regulatory agencies. He currently works for Roche as a global statistical and regulatory consultant within the Statistical Methods and Research Group, and in this role he works across all therapeutic areas and phases of drug development. So he's a former president of the International Society for Clinical Biostatistics. He's joint editor of the Journal of the Royal Statistical Society and associate editor of Statistics in Medicine, Pharmaceutical Statistics, and the British Journal of Clinical Pharmacology. He's published widely, including being the author of one book called Dictionary for Clinical Trials, this rivals Kurtz, and is joint editor of the Textbook of Clinical Trials, both published by Wiley. So in addition to his other responsibilities, he's lectured wildly and widely, right, for different organizations, including the London School of Hygiene and Tropical Medicine, and also has given courses at our FDA on the science of small clinical trials. But he's made also many contributions to our curriculum at Johns Hopkins. Since 2007, he's co-taught with Dr. Mary Folks one of the online courses offered through the Department of Biostatistics entitled Biostatistics in Medical Product Regulation. In fact, this course is going on right now, and if you are a student who would like to add it, you can do so as of 11:59 p.m. today. And lastly, he and Dr. Folks are currently developing a course, a brand new course. They gave it first in the Summer Institute, and they're developing for online offering this coming third term called Non-Inferiority and Equivalence Clinical Trials. And it will be offered, as I said, during third term. So it's my pleasure to welcome Dr. Simon Day.
Thank you very much. Well, thank you for that wild introduction. How do I live up to that? Good morning. Thank you all for coming. It's a great coincidence and pleasure for me to be here. Kay Dickerson said to me a while ago, "Oh, when you're passing through, you must give a seminar." Well, you know, I live in London. I don't pass through Baltimore that often. And then she emailed me a while ago saying, "Hey, how about one of these two dates?" And it just so happened that yesterday I had to be here in Baltimore. Later today, I have to be down the road in DC, and I had nothing to do this morning. And so it actually came together really very conveniently. So I'm very pleased to be here. Thank you for the introduction and the invitation. What constitutes reliable evidence? Well, lots of different things. This talk I've put together over a number of years. I just want to motivate it by thinking about patients, because that's what this whole business is all about. This is a picture of a little girl, Sasha Brown. The fantastic thing about this picture, if you were her parents, is that it was on the front page of the Sunday Times, a really quite a large picture. The parents, the grandparents, they will have bought copies of these and shown it to all of their friends and relatives. It's a beautiful picture, I think. I have to say, if it were my daughter, I know my wife would have been straight in there with a hairbrush. But that aside, what a wonderful picture it is. But there is some happiness and some sadness to the story. Sasha Brown has taken part in the first trial, at least that's what it was described as, the first trial to treat cerebral palsy using stem cells taken at birth from the patient's umbilical cord. Her parents say her condition has improved. Well, and I hope it has. Whether it's improved because of the stem cell therapy is another question. And I'd like you to think about Sasha over the next 50 minutes or so as we go through this. Or perhaps other little Sashas that you may know. So think about patients. There's an interesting organisation called ICORD, which is the International Conference on Rare Diseases. It's something that I go to regularly when I can. It's an interesting mix of science, policy, patient organisations and so on, all coming together to try and tackle the problem of helping patients with rare diseases and indeed helping their families and so on. And at the end of one meeting in 2007, somebody, it was almost a closing comment, somebody from the audience said, we need to stop always thinking about evidence-based medicine. And I was about to jump up and say, no, 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 we must think about evidence-based medicine. And it just occurred to me that actually this might have been a parent or someone from a patient organisation, and it might be a little bit rude of me to jump up like that. So I kept quiet, but I thought about it a lot. And I went back the following year to the 2008 meeting, which actually was in Washington, D.C., and that's where many of these ideas really started to stem from. Here's an example of convincing evidence. Published in the BMJ, parachute use to prevent death and major trauma related to gravitational challenge, a systematic review of randomized controlled trials. So they published very clearly what their systematic review criteria were, what their search criteria were. They then carried out this search on pre-specified databases, and they found no randomized controlled trials. But they still came to the conclusion that using a parachute was probably a good thing. Now, this is not a laughing matter. Certainly not if you're randomized to placebo. Okay. So they wrote this somewhat tongue-in-cheek, obviously. But the point that they were making was you don't always necessarily need a randomized controlled trial to fairly reliably get an answer to a question. This paper generated an enormous amount of correspondence on the BMJ website, just the rapid responses, including, for example, examples that even with a parachute, some people sometimes have major injuries or die, and without parachutes, sometimes people still survive. So yeah, it's not a one-to-one relationship, but all the same, we feel reasonably confident that using a parachute is a good thing. One of the correspondents to this was Bob Temple, and I have to say I read this paper, and as a regulator I thought, no, I just keep my head down, keep out of this. Yeah. But he waded in, and he pointed out that the Code of Federal Regulations describes various possible types of control groups that might be reasonable, including historical controls. So he was suggesting that there's nothing wrong with this at all, and it fits quite well into what CFR says, and indeed what ICH says, and so on. So sometimes we might get away with historical controls. Well, Tom Chalmers very politely wrote this short article in 1968 asking when should randomization begin. I say quite politely because he knew the answer, and actually this short article asking when, the purpose was to state it should start right from the very outset. The very first patient should be in randomized trials. He went on, 8 years or so later, stating a bit more firmly, randomization of the first patient. And eventually he seemed to have got rather cross about this, and randomize the first patient, ! Do it. And I feel very strongly about this, that we do need to randomize from a very, very early stage. Bradford Hill, one of my fellow countrymen, of course, he wrote that frequently we have no scientific evidence that a particular treatment will benefit the patients, and we are often willy-nilly, wonderful expression, we're willy-nilly experimenting upon them. It may well be unethical not to institute a proper trial. So in the concept of rare diseases and serious diseases, there's a big push to try and get treatments out to patients. And of course, I want patients to get treatments if they're good and safe treatments. But do we want to delay things by running randomized trials? And I'd just ask you to think about Sasha. She was not put into a randomized trial. She was just given this treatment as a hope. And did it do her any good or didn't it? We don't know. Her parents think that she's improved. So if you're the next parent down the line with your child, what do you now think? Think, well, I don't know, but it's worth giving it a go. And then we get into the situation that it becomes impossible to do randomized trials. Patients and many investigators just aren't prepared to randomize patients. Some commentary on this idea of randomize the first patient. Spodek says, it's always possible to do a randomized trial. I've slightly paraphrased what he said, but it's pretty much always possible in the search for a real answer, and it ensures an ethical approach that gives every patient a 50-50, well, it doesn't have to be 50-50, but every patient a 50-50 chance to get the best treatment. That is, and I emphasize it here, not to get the new medicine, not to get it at a time when its effects and risk-benefit are not understood. So my way of thinking about this is patients who volunteer to take a potential new medicine at a very early stage of its development actually have the right to have a reasonable chance of getting placebo. And of course, the push for this is, I might get the treatment, I might get the treatment. But I think they should have a reasonable right that actually, if they're lucky, they might not. I just picked up this cartoon from somewhere. There's the guard and the prisoner. I would point out, I do sit on an ethics committee. I don't think we've ever had an application to do a research project on prisoners. But would you like to volunteer to test a new medicine, says the guard. Well, only if I get the placebo, says the prisoner. And I don't blame him. I don't know who this person is, Con, but in an article in Clinics in Gastroenterology, he said that he would hope to meet the inclusion criteria for controlled trial, enter the trial, and then refuse treatment. Now, I don't advocate this. Please don't do that. But you can see his point. You get looked after well in clinical trials. It's not to say you necessarily want the treatment. Here's some stuff that you may be familiar with. When I give talks like this outside of Europe, people tend not to do it. But a proposal to amend the Food, Drug and Cosmetic Act to create a new 3-tiered approval system for drugs, biological products and devices that is responsive to the needs of seriously ill patients. I've never understood what this last phrase means, well, and for other purposes. Seems a wide catch-all. But this was an idea that as soon as products have at least gone through some early phase 1 safety studies, then if there's a vague hope that they might be useful, just give it to patients. And I can see the logic on the first level for this. What's the alternative? The alternative might well be, actually, there's nothing. So I can see the thinking behind this. But I think it seriously leads to damaging the possibility of carrying out proper controlled trials and then never being able to properly answer the question of whether these therapies work. And I know the Society of Clinical Trials wrote a fairly strong response pointing out the weaknesses of this, and I would strongly support that statement. Part of my evidence for supporting that statement, and indeed it was highlighted to some extent in the Society for Clinical Trials response, is how good are we, and by we I mean the pharmaceutical industry. I suspect I'm the only one in the room that works for the pharmaceutical industry. So how good am I at developing new drugs? I'm sorry to say, really not very good at it at all. I've been doing it for a long time, and I'm still not very good at it. It's quite a difficult thing to do, but these are some data about the attrition rates of new hopeful drugs as we go through phase 2, 3, and on to registration and approval. So this is after drugs have gone through some phase 1 testing, some acute toxicity testing. And you can see that about 90% of drugs don't make it. That's why I say we're really not very good at doing this. So why don't they make it? Well, partly it might be the design of trials, but here's a comment about the dramatic unpredictability of single-arm, uncontrolled phase 2 trials. So that's one reason why in oncology, I think a lot of drugs fall out at phase 3, because we delude ourselves with their efficacy in phase 2. But here's some data about why drugs drop out of the pipeline. And with an interest in rare diseases and orphan drugs, I could say it perhaps saddens me a little bit if there's some potentially good drugs out there that companies decide to drop the development of for economic reasons. These may be very good, valuable treatments for patients, but the manufacturers decide there's not enough money in it, so I'm going to get out. Now, the decision is more complex than that, but as I say, that would sadden me somewhat. But even putting that aside, the reason that most drugs fall by the wayside is either because they don't work or they're not safe. Well, these two things are much related to each other. There's plenty of very unsafe medicines on the market, and they're there because they treat very serious diseases. They may have nasty side effects, but the benefits outweigh the risks. But from the early 90s to the late 90s, early on about half of drugs dropped out because the benefits didn't outweigh the risks, and that seems to have increased a little bit. Here's another publication a bit later on, looking at 1991 and 2000. And again, there's the commercial reasons and there's the cost of goods. So it would sadden me somewhat if potentially good medicines weren't making it to the market because of those commercial reasons. But again, putting that aside, we've got toxicity, which is safety. We've got clinical safety, and we've got efficacy. So 52% in 1991, 61% in 2000 have fallen out because the efficacy isn't good enough to outweigh the safety issues. So the benefit risk is not positive. Here's the most recent example of this type of thing I've found, published just a month or two ago. And again, up the axis here, we've got attrition rates. And phase 1, 2, and 3, we've even got preclinical here. So we expect high attrition rates early on and hopefully lower further down. But even so, we seem to be getting worse. In 2004, the attrition rate at phase 3 was up at around 60%. In 1990, it was down at about 25%. So huge number of drugs don't make it through the pipeline.
What do you mean by attrition? What's the meaning of attrition rate there?
Essentially a drug that perhaps went into phase 2 trials and then never went any further. Now, I'm assuming that that's because it didn't work in phase 2. It could be other reasons, commercial reasons as well for that matter, but essentially drugs that stopped at phase 2. So back to this randomise the first patient idea, given that actually most drugs don't work, most drugs fall out because the efficacy doesn't outweigh the benefits. Patients who volunteer to take a potential new medicine, and that word potential is there intentionally, at a very early stage deserve the right to have a reasonable chance of getting placebo. Most of these sadly don't work, and they have a negative benefit risk. So you, your loved one, Sasha perhaps, might be better off on placebo than taking this experimental treatment. It's a sobering thought. I just slotted this in fairly recently. It struck me as something quite poignant. Bayesians in clinical trials asleep at the switch. didn't entirely understand why that title, but it's a bit catchy. And a couple of comments. It's difficult for physicians, and I would argue others too, to keep in mind how bad things may be with an untested intervention in the face of the reality of how bad things are without it. And I think that's very true. You know, we are desperate for new medicines in at least some therapeutic areas. And we can see how bad things are without these medicines. Surely it couldn't get any worse with a new medicine. Well, it's inevitable that with a new medicine there will be some side effects, and it may have no therapeutic benefit at all. So actually it could be worse. So that's quite a sobering thought. And also with regards to thinking about priors on our treatment effects, the more passionate the investigator, or perhaps the patient or the parent or somebody else, the greater the protection the priors require from their strongly held opinion. So if it's your child, if it's my child, you know, how passionate are we about the possibility of a treatment for a disease that otherwise has no treatments? We're very, very passionate. And it's very difficult to then perhaps come down to earth and say, well, actually, is it really going to be beneficial? Particularly when I was a regulator, but I still see it now, lots of arguments against doing very small trials. We can't do a randomized trial because we haven't got enough patients. If you've got 2 patients and a coin, we can do randomized trials. We haven't got any degrees of freedom left for error term, but okay, 3 patients and a coin. So it is possible to do randomized trials. There might be other constraints, but simply lack of availability of patients is just not a winner. Related to this, there's no point having a control group because the trial would be severely underpowered. And no point having a control group because there's no chance to show any treatment benefit. Well, that's a bit ironic, isn't it? But I've heard that from regulators and from applicants too. The control group isn't worth it. Well, is that true? This is just a hypothetical example, but putting some numbers on it actually does put it in perspective a bit. Let's say I'm looking at a treatment that's pretty much has no cure, so I'm saying that in a trial we might get about 10% response, a bit of regression to the mean and things like that. And our new treatment perhaps has quite a substantial effect. We get a 50% response. If we have 2 equal-sized groups of 20 patients, we've got about 85% power, at least for a one-sided test of 5%. Well, that's not bad. So there's 20 patients in each group, there's 85% power. What if the control group gets smaller and smaller? So we keep 20 patients treated with this new proposed hopeful treatment, but the control group gets smaller and smaller and smaller. Well, the power drops off. What if the control group gets smaller still and goes down to 0 patients? Well, the power drops off even further, goes down to 0. So I'm sorry, but your study without a control group has 0 power to detect any treatment effect of any magnitude at all. If you let me randomize at least 2 patients to a control group, at least I get 20% power, which is a lot more than you've got. I've tried this. It don't work. The answer comes back, oh, well, look, young man, I would treat all of those 20 patients, or maybe if we're going to budget for 22, because I wanted 2 controls, maybe all 22, with the active treatment. I don't know where they get the word active from, but that's their belief. And I'd be able to compare them to historical controls, you see. Got the answer to that one too. My small trial has 20% power, and I can compare to historical controls as well. So what am I losing by having at least some sort of small control group? And in fact, if we went for two equal-sized groups, if we said we're going to expose 20 patients to this trial, and we put 10 in each group, or maybe it's 22 patients in the trial, we put 11, we've still got about 50% power. So actually, there's a lot that can be got from randomized controlled trials, even if they're very small.
But you still lost the argument.
Can I take a vote of hands? Have I lost the argument? Sometimes yes, and sometimes no. I think very often I've managed to persuade people that they should do randomized trials.
Are you open to adaptive randomized trials? Or here you design a very simple one?
I've designed a very simple one. Anything that we do that might be more sophisticated is probably going to add even more benefit. So even starting with the absolute simplest of randomized controlled trials, we can still get something out of it. And maybe we could do more with some sort of an adaptive design. Why this dichotomy between historical controls and concurrent controls? It seems a bit strange. And here's a paper by Stuart Pocock. It was written in 1975. How old was I then? Ooh. Still had short trousers. and I kind of call it beige without computers. But he went through the common situations that you might meet and how to combine randomized and historical controls. So this stuff's been around for ages, but I don't actually see people doing it very often, including myself, I suppose I should say. Back to this idea of randomise the first patient, even small trials are not completely useless, as I've tried to suggest to you. And just a few examples. Ken McCrae, very flamboyant statistician, sadly no longer with us, but he wrote about the value of small clinical trials. John Matthews, who is still with us. Small clinical trials, are they all bad? Well, with a title like that, you can almost guess what the answer is going to be. So no was his answer. They're not all bad. There are problems with them and deficiencies. But what's the alternative? If the alternative is no trial, maybe this is a good thing. Let's go to the other extreme, the large mega trials, the sort of thing that Richard Peto advocates and gets involved in. 20,000 patients, well, that's just a small one, obviously. 30,000, 40,000 patients, trials of this size. Why do they do trials that are that big? Richard Peto can do power calculations, and it doesn't come out this sort of size. But the reason he does them is because he looks at the existing evidence base and says that the only thing that will substantially add to that evidence base is a study of this type of size. If I, as Richard Peto, do another study of aspirin of 5,000 patients, it's not going to impact on the current evidence base at all. It's just not worth doing. So I look at this idea and think, what do we need to do in order to move science forward, in order to move forward our understanding of a potential new therapy? And I think a new study should usefully add to the evidence base. If it doesn't, you really have to question the ethics of it. If there's a lot of evidence, new studies have to be big. If there's very little evidence, then actually even very small studies might still substantially add to our evidence base. We might prefer even larger studies, but if there's difficulties with doing those, perhaps the rarity of the patients or even other reasons, then small trials might still add to the evidence base. And there's a nice example here, David Machin, who I worked with, strategy for randomized trials in rare cancers. So they talk about this kind of idea. And from the economics literature, the economics of more research is needed. That wonderful phrase that comes at the end of every paper that we write in order to help justify the next grant application. But when actually does it make sense economically to do new studies? So that's an interesting paper to look at. A bit of a caveat here, how much evidence exists isn't the same as what's the current sample size. It's often related to it, but large studies of poor quality may mean that we have a fairly weak evidence base. So it's not just a question of how many patients already treated. And I'd also say I think there's an ethical issue about doing a very small study when a usefully bigger one could be done. So there's an argument for collaboration between centres, which I know can make trials more difficult, but if the sample size is 12 because that's all I can get in my clinic, I think there's possibly a problem making that justification. So get together with other people and do these things. Okay, moving track of thought a little bit. Levels of evidence. Well, at the CHMP, which is the European regulatory agency, where I did some work for 5 years, we produced a document called Guideline on Clinical Trials in Small Populations. I was the lead author for that guideline. I have to say I don't entirely like it. These things are written by committees. But I think it actually was quite a good start, and I've talked to them and tried to get them to update it, because I think there's been a lot of thinking since 2006. I think actually it should be about samples, not populations, and I think it shouldn't be about clinical trials. It should be about studies. So apart from trashing the title, there's various issues in the content that I don't entirely like. But it was a start. Within this document, really in the introductory comments, we listed this hierarchy of evidence. And in particular, we changed the first one between a fairly late draft and the final draft to say not meta-analyses, but meta-analyses of good quality randomized controlled trials. There seems almost a myth that if you say, I've done a meta-analysis, everybody says, okay then. But actually it needs to be good meta-analyses of good randomized controlled trials. So we put that at the top and then going down this. Right down to the bottom, this is not of no use at all, opinions of experts in the field, but it is probably the weakest evidence level that there is. It's just worth pointing out that the group of people who wrote this guideline were nothing more than the opinions of experts in the field. Perhaps if we'd sat in a field, it might have been more interesting. But actually, we can go back something like 20 years. Sylvan Green, who was a good friend of mine, anecdotal case. He basically did the same thing the other way up. So anecdotal case reports as the worst, but not zero level of evidence, down to, well, in 1984, they referred to confirmed randomized trials. I suspect if they rewrote it today, they'd have used the term meta-analysis down there. But essentially the same thing. But none of us have really invented much. Let's go back another 20 years. And I was around then, but don't even think I was in shorts then. This is Bradford Hill. I've already quoted from him. He wrote wonderful stuff. And this is in the context of observational epidemiology, which actually is the same thing as an uncontrolled trial. And he wrote about strength of association, consistency of effect. Do you get the same effect every time you do this? Specificity, temporal relationship, and so on. And these are often called the Bradford Hill criteria. You may have heard of that. But they're not a bunch of criteria. And he said that none of my 9 viewpoints, that's what he called them, not criteria, can bring indisputable evidence for or against the cause and effect hypothesis. So what we're interested in is, did this drug cause this benefit? Think about Sasha. Did the use of the stem cell infusion cause the benefits that have been perceived? So none of them are absolutely required. What they can do with greater or lesser strength is to help make up our minds on the fundamental question, is there any other way of explaining what has happened? Explaining perhaps the response that Sasha seems to have received. He went on to say, what I do not believe, and this has been suggested, is that we can usefully lay down hard and fast rules of evidence that must be obeyed before we accept cause and effect. And this is the thing that I think gets forgotten. It's not a tick box approach, and although I generally am very hesitant about uncontrolled studies, as perhaps you've gathered, there are some situations where maybe it might be acceptable. So I don't want to be randomised to placebo if you're getting the parachute. Okay. Don't even want to be in a crossover trial. I hesitate to criticise the Cochrane collaboration, but these are their ideas of levels of evidence, and in general I would agree with those, and I just summarise them there. Essentially, level 1 is randomised controlled trials, level 2 is case control studies and cohorts, and level 4 is expert committees writing guidelines and things like that. So levels of evidence. Then they've got strength of recommendation or grades of recommendation. And again, I'll just simplify that a bit. Grade A recommendation about use of a treatment, or perhaps actually not using a treatment, comes from the higher level standards of evidence and down here. So we've got this reasonably obvious relationship. Levels of evidence might be higher or lower. Grades of recommendation will be higher or lower. And the higher the level of evidence, the stronger a recommendation can be made. And it goes something like that. But I think the thing that's wrong is this bit here. That link needs to be broken and put in context. It's not the same link in every situation. Think about parachutes if you want to. So I think what we need to do is think about context-specific evidence. So rather than just go to the menu that says highest level of evidence is start with randomised controlled trials and then move on to meta-analyses, think about it in context. And if we have, for example, a stable disease, a chronic disease, and we intervene at some time point and the response goes like this, that's fairly convincing. If we've got a fluctuating disease, so I don't know when it's going to go up and down, and again, we intervene in the same way, and that happens, that's pretty convincing. Not quite as convincing, because you don't know if perhaps we were just about to come down, and we seem to be lucky and have come down quite a long way, but not quite as convincing as that one, but it still seems reasonably convincing. If we've got a fluctuating disease and the treatment benefits us, but all it does is reduces the symptoms or reduces the intensity, that's not as convincing. We really don't know if this green dotted line is where the natural course of the disease was going to go or whether it was going to go somewhere else. And then if we've got something like migraine, perhaps, that happens in episodes and then goes away, and at some unknown time in the future it will happen again, or epilepsy or something like that. And I intervene and I actually prevent that spike. I prevent that spike, but of course I don't know I prevented them. I just didn't see those spikes happen, and I don't know if the patient would have had that spike or not. And this becomes particularly unconvincing, particularly so. And so I'd ask you to think about Sasha, and I think that she probably fits in this category. Her symptoms are better or worse from one day to another or from 1 month to another. We have intervened at some particular point in time. Her symptoms haven't gone away. So did we actually have any effect, or is this the natural course of the disease? We really have absolutely no idea at all. Getting back to my passion for rare diseases, this is an interesting concept to think about, and I I'm not sufficiently biologically educated to be able to justify this. But the idea that rare diseases and serious diseases, which of course are not the same thing, but they do very often overlap, and dramatic treatment effects, which sadly we don't see as often as we might like, but we do in some situations. And this link, or this overlap in the middle here, this idea that uncommon tumours and exceptional therapies, is it a paradox or is it a paradigm? And the argument that these authors put forward is that if a disease is very rare, it may well be that it has rather few causes, and that's why it's rare. And so all you've got to do when you're treating this patient is tap off that cause, and it goes away. Now, it's only an idea, but if a disease has lots and lots of different causes, you can see the logic that it's quite likely there may be more patients around with this disease because there's lots of different ways of getting it, and having a treatment that taps one off doesn't really have a lot of impact. So I follow the line of thinking, and I'm sure that biologists and perhaps pharmacologists would have a field day discussing this. I think picking up on that point, Bruce Alberts, who certainly was, I don't know if he still is, editor-in-chief of Science, if I were the czar of cancer research, I'd give high priority to recruiting great scientists to decipher the detailed mechanisms of apoptosis, cell death, and DNA repair. You know, get at the root cause. That's what he's arguing. And of course, great idea. I guess we'd all agree, but maybe it's not so easy to do. So to begin to wrap up, do we need to stop always thinking about evidence-based medicine? Well, my strong and I would say really quite passionate belief is no, we shouldn't stop thinking about evidence-based medicine. But we need to think very critically about what constitutes evidence and in a particular scenario. So I think if this lady at the ICORD meeting in Brussels had said, we need to stop always thinking about randomised controlled trials, then I'd have still got impassioned and thought about it, but I fundamentally would have agreed with her. But I think we do need evidence-based medicine because patients deserve it. But we need to think about what evidence we need and what constitutes the best evidence. And what I would say strongly is this is not a means to think, what's the minimum we can get away with? It's a means to think, what's the minimum that we need to make a good decision? So a subtle but important difference between those. And I think that enough evidence in one setting may not necessarily be enough in another. And the ethical argument of last chance therapy, just give it to patients, they've got nothing to lose. Actually, they have. And I think that's a disastrous way of thinking, really. I think it's bad both from an individual patient basis, because, as I've told you, most of these therapies actually have a negative benefit-risk balance. So it's bad news for patients, and it's also very bad news for drug development and getting good evidence about therapies. So I just end with this last slide, thinking back to the parachutes. What causes death? Well, I think we all know it's the speed of hitting the Earth. Don't need a great scientist to work that out. But we do know the mechanism of action, that parachutes slow you down, so they probably help. And if we're lucky, we might have a happy and safe landing. Thank you very much.
We have a few minutes in which you can ask some wild questions. So I'd like to open this up for discussion.
How long did it take you to do that last slide?
About half a day, actually. But it was worth it, wasn't it? Do you want to see it again? Yeah.
Yeah.
I mean, I really like your viewpoint, and for me, releases, at least if people listen to you carefully, it releases from what I'll call the tyranny of these hierarchies. And I don't mean that I don't believe in the order of the evidence in some abstract sense, but I do think the idea of needing to dock it to context and to not just evidence, but what the evidence, not just quality of evidence, but what the degree of evidence is in terms of separation, absolutely needs to be there, even though it sort of is an invitation to ride fast and loose. I think with at least skilled scientists and clinicians working together, you've kind of broken that tyranny a little bit, and I like that.
Well, thank you. I think that tyranny is there. Giving talks like this to uninitiated audiences, so I'm okay here, but to uninitiated audiences does seem to give them a license to say.
Barbara out of the room.
I'm not leaving the country.
Does seem to give a license to say, well, actually, we don't always need randomized controlled trials. And I think we don't always. But I think usually we do. And I think many of the arguments that people use of why we can't, it's unethical. As Brad Vittil says, it may well be unethical not to do a randomized controlled trial. Can't do it, you haven't got enough patients. That's just ridiculous. I look at bioequivalence studies quite often. 16 healthy volunteers. We have regulatory guidance that says thou shalt not do a bioequivalence study with less than 12 subjects. Okay, so they're happy with 12. So you can do, and that's 12 in a crossover. You can do small studies and get pretty reliable evidence in some situations.
Sort of a related but maybe slightly different question. Right? This is great, thank you. I've recently been, so at National Institute of Mental Health, there's been a recent push against pilot studies in some sense. Well, I shouldn't say against using pilot studies to inform whether a larger trial should be done. Elena Kramer wrote this very influential paper in the mental health world, and it's gotten a lot of attention. I'm just wondering what your sort of take on that is, because the argument is that the results you get from a small study are so variable that it's not actually very informative as to whether,
Yeah. I've been asked that question before, and so I've thought about it and struggled to come to a good answer. I very much dislike seeing studies proposed as a pilot study when it's quite clear to me the reason it's a pilot study is because we can't be bothered to do a bigger study. And that, I think, fits in with what you're saying. When I see a pilot study, I always ask to see the draft protocol for the study. So we can see, and of course I never see it, but the point being that we can see what is this pilot study going to do? What's it going to give us to help us do the study? And it might be something as reasonably trivial as estimating variability. Now, you might argue you could do that as an internal pilot study. It may well be, and it ought to be, can we actually get patients through the clinic to do all these tests in that time that we thought we were going to do? That's what pilot studies really should be about, is the practical issues. So I think I'm agreeing and disagreeing. I think if pilot studies are done for the right reasons, then they're very valuable and potentially can save an enormous amount of resource and patient exposure to a trial that isn't going to work. That's not a statement about the treatment, it's about the trial. So I guess we probably have similar views on that. Okay.
Yeah, back to Tom Chalmers and randomized from the first patient on, which it seems, in principle is a good idea, but practically it's almost impossible with the paraphernalia you need for randomization and so on. So any thoughts or comments as to how to bridge those two ideas into one?
Well, are you talking about the technical difficulties of randomization or the procedural and regulatory difficulties of doing a randomized controlled trial?
I've got to do the dance in front of an IRB, and I've got to establish that blah blah blah. And so on.
Yeah. So it's not the technical difficulties; it's all the paperwork.
Exactly.
yeah. No, I agree. I sympathise with that a lot. I think what we need, we need good clinical practice or good clinical research practice, whatever you want to call it. But again, context specific. And I fear that GCP has turned itself into a nice little industry, employing lots of people, allowing lots of courses to be run, which further employ lots of people. I don't mean to sound too cynical about it, but I don't want trials to be done badly and where we can't rely on the data that we get from them. And so I think we need a compromise on GCP. But I have to say, unfortunately, I don't know where that compromise is. And I'll just give you an anecdote. When I first joined Roche about 4.5 years or so ago, I was at just an internal lunchtime seminar that somebody was giving. And it was hedging on GCP and issues that we need to do. And somebody, perhaps they were new to the industry, I don't know, but somebody asked, why do we have to do all these things? And the answer from the speaker was because the regulators tell us. And I said, no, that's not why we do them. We do them for our benefit. How many times have we, whether that be you people here or us at Roche or wherever else I've worked, have tried to go back to the records of that trial that we did a few years ago because an important question's come up. And the guy that did it has left, but we think that they contracted out to somebody. He wasn't actually a statistician, he was a psychiatrist, but he'd done some statistics training. You know, you get this sort of stuff, and that's why if we contract out, we want someone's CV, we want to know they're qualified to do what they do, and all the other things too. So it's very easy in any situation to say, we don't need that for this trial. But these things sometimes come back and bite you some years later. So I would love us to be able to find a compromise. And it's not simply academic versus industry. As academia and industry work more and more closely together, it all becomes very blurred.
Really enjoyed your talk. But as I was listening to it, you really were focusing on one type of trials that test medications, with the exception of parachute, which is sort of. And the areas that I deal with are prevention, where you're dealing with contrast that might be, the exposures that might be decades long. And the other area is technology, where the treatment or exposure in this case is evolving rapidly over time. So rapidly that by the time you decide to go in the field, you're almost dealing with something obsolete. So I was wondering how you deal with this. I often have thought that when we decide whether or not to do a randomized trial, we actually, and we decide not to, and I think there are many instances where it makes sense, where there's a process, though, where you go through step by step and say, we just can't test smoking on total mortality because we can't establish the contrast and maintain it over time. So, your thoughts?
That's a very good point. And you're right, I have really just focused on trials of pharmaceutical products. A halfway house, perhaps, between what I've talked about and what you're talking about might be medical devices, which also move on fairly rapidly. I think a nice aspect of device trials is that as the device develops, you've got a pretty good idea what it's going to do because you used version 3 last year. And I think this is why, I haven't explicitly asked them, but I think this is why the FDA was happy to write their guidance about the use of Bayesian approaches in device trials. And they have, I know they have had successful applications where they've used informative priors, so not just vague flat priors, but informative priors and then supplemented them with data. Because if you've got a device and you change the way some of the wiring or one of the springs works, you've got a pretty good idea what it's going to do. And then you've got to just test out whether it does. Unlike a drug where, I'll just change an atom here and looks pretty much the same to me. Well, yeah, it might do anything. So that's a halfway house, and again, randomized controlled trials may not always be necessary there. Moving further towards what you're talking about, we need some good thinking, because some of these situations that I think you might be talking about, I might argue, need randomized controlled trials, but we just can't do them. I mean, smoking and lung cancer, for example, would be a classic situation. Perhaps we don't need them now, but many years ago we might have done. So I don't. So what's the answer? You must know the answer. You've thought about this.
The one that comes up is salt and total mortality.
Right. Okay.
And, I've done many trials with salt and blood pressure, and there's a strong relationship. But then there are naysayers who can always come up with a biologically plausible reason why it could be harmful. But it'll never be done because you can't establish the contrast maintained over time. Or you spend $10 billion to do a trial where there's a high risk of a false negative. So I don't know the way out of that.
No, okay. And another linked example is questions that are important to know the answer to, but which, even though you might be able to do a trial, by the time you got the answer, the question would then be irrelevant because therapeutics would have moved on.
But I think my point is that there needs to be some sort of thinking process about which trials can be done, and then explicitly documenting when they can't be done as reasonable.
Yes. No, I think that's true. That the one caution I would give is can't do doesn't equate to don't need to. Doesn't equate to therefore something else will be all right. If there's something else, like in the parachutes, will be enough evidence, that's fine. But just saying, well, it'll be all right to do this because we can't do a randomized trial, there's a break there.
Well, I hate to bring this discussion to a close. It's been a fantastic morning, but I am cognizant that, unlike what that clock says, it's after 9:30. So please join me in thanking Dr. Day.
Thank you very much. Thank you.